Today we are going to talk about RNA and protein synthesis. Genes are portions of DNA that control the production of proteins. Genetic messages can be decoded by copying part of the nucleotide sequence from DNA into RNA, and RNA contains coded information for making proteins. There are four main differences between RNA and DNA. Three of them we have already discussed. The sugar in RNA is ribose instead of deoxyribose. RNA is single-stranded instead of being double-stranded. RNA contains uracil instead of thymine. And DNA stays in the nucleus while RNA can leave the nucleus and go into the cytoplasm. There are three main types of RNA, and they are all responsible for making proteins: messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA carries copies of instructions for assembling amino acids into proteins. Remember, messenger RNA is made in the nucleus. Ribosomes are made up of proteins and ribosomal RNA. Ribosomes are floating freely in the cytoplasm as well as attached to the endoplasmic reticulum. During protein construction, transfer RNA transfers each amino acid to the ribosome. At the top you have the amino acid attached, and at the bottom you will find nucleotides called anticodons. These pair up with the codons on messenger RNA. There are only 20 known amino acids. Of the 20 amino acids in your body's proteins, 9 are essential to your diet because your body cannot make them. That's why it's important to supplement or eat foods that are high in these particular amino acids. The genetic code is the language of mRNA instructions. The code is written using 4 letters: A for adenine, U for uracil, C for cytosine, and G for guanine. Protein synthesis involves 2 processes: transcription and translation. Protein synthesis begins in the nucleus with a process called transcription. DNA is copied in the form of a single strand of RNA, messenger RNA. The process begins at a section of DNA called a promoter. An enzyme, RNA polymerase, acts on the DNA molecule and causes it to unzip, basically breaking those hydrogen bonds between the bases. It chemically tells RNA nucleotides to come and base pair with the open DNA molecule according to the base pairing rules. Adenine pairs with uracil in RNA, and cytosine pairs with guanine. This is basically a picture of transcription. The blue portion represents the DNA molecule, the open DNA molecule, and this red portion represents this new RNA molecule, this new mRNA molecule being made. Remember that in DNA you have adenine, but it's going to pair up with uracil in RNA. Remember, there is no thymine in RNA. RNA editing. Some DNA within a gene is not needed to produce a protein. Those are called introns. Exons are portions of the gene or base pairs that are used to make proteins. Introns are portions of the gene that do not code for protein synthesis. I have attached a little video clip that shows you the process of splicing. Splicing is removing those introns from pre-messenger RNA before it is released into the cytoplasm. Remember, DNA stays in the nucleus, but the edited messenger RNA leaves the nucleus and goes into the cytoplasm to the ribosome. Translation. Translation is the decoding of a mRNA message into a polypeptide chain, basically a protein. Translation takes place on the ribosomes, whereas transcription takes place in the nucleus. During translation, the cell uses information from messenger RNA to produce proteins. So this is a picture of the nucleus. It's showing you how messenger RNA is leaving the nucleus and moving to the ribosome to begin to start making these polypeptide chains. A codon consists of 3 consecutive nucleotides on messenger RNA that specify for a particular amino acid. So every 3 nucleotides on mRNA make up a codon. AUG is a codon, AAC is a codon. The ribosome binds to new tRNA molecules and amino acid as it moves along the mRNA by matching tRNA anticodons to mRNA codons according to base pairing rules. So what happens is messenger RNA leaves the nucleus. It goes out into the cytoplasm to pair up with the ribosome. This big blue and tannish blob, that is the ribosome. The ribosome is going to bring tRNA over to connect the anticodon with the correct codon located on messenger RNA. So for instance, we have a codon here, AAA. What is the complementary base pair to adenine in RNA? It is uracil. So we have the correct anticodon here. Transfer RNA will come and connect with the AAA, the codon on messenger RNA, and this amino acid will be released. As the amino acids are brought close together, they are joined by peptide bonds to make a protein. So when this anticodon pairs up with the correct codon, this amino acid is released and a peptide bond will begin to form between these amino acids, eventually forming a long polypeptide chain. The process continues until the ribosome reaches a stop codon. So here you see we have our mRNA transcript. This is the ribosome. This is tRNA that's leaving because it doesn't have an amino acid anymore. Remember, when the anticodon pairs up with the correct codon, the amino acid is released. A peptide bond forms between these amino acids, eventually forming a polypeptide chain, which is a protein. This is just a picture showing you transcription and translation. First, information is copied from DNA. That information that is copied from DNA is now our messenger RNA. Messenger RNA leaves the nucleus, goes into the cytoplasm to pair up with tRNA to produce these amino acids. This chain of amino acid is going to produce our protein. This is just another diagram showing the same thing. This is the portion of DNA that is being transcribed. So we have our DNA strand. This information is copied from DNA, mRNA. mRNA leaves the nucleus, goes into the cytoplasm to pair up with tRNA to release these amino acids that will eventually form a protein. Alright, so this is a genetic code. We could use this to determine what kind of amino acid is going to be produced. If I gave you a codon, you should be able to take that codon and tell me what kind of amino acid will be made. This is another chart showing the same thing. If I give you a particular codon, you can use this chart to tell me what type of amino acid will be made. And I will show you how to use both of these in class. So we are finished. If you have any questions, shoot me a message through Remind or send me an email. Have a good one.